Now, having made it quite clear that the role of the senses in perception is not simple and that the senses should not be considered as simply five kinds of input channels, we need to explore this area a little bit. We noted that taste and smell are very closely linked. We noted that vision and the vestibular system in the middle ear work closely together to orient us with respect to the world. And we noted how the many receptors that give rise to proprioception, kinesthesia, and interoception give us a sense of awareness of the state of the body, its position in space, its movement through space. So with all this, we're now going to turn to three modalities. We're going to treat them as modalities. We're going to talk in particular about these three: vision, audition, and touch, or sight, hearing, and touch. And we're going to note that these have a very special relation to the central nervous system, the brain, if you will. And that this relationship is evident in the manner in which the anatomy is revealed. Each of these three sensory modalities, considered now as sensory modalities, noting that this is not a simple matter of receptors, each of these modalities has a primary sensory surface. That's the retina for vision, the basilar membrane in the ear for audition, and the skin for touch. And there's a corresponding primary sensory cortex, and that we can identify the relationship between the sensory surface and the layout of the primary sensory cortex. This is a topological argument. We're looking at the spatial relations between specialized surfaces on towards the exterior of the body and the manner in which they map to the first part of the cortex. So the retina is the skin at the back of the eyeball. That's where optical texture, changing patterns of light and dark, are converted to interplay with the activity of the nervous system. Now, the primary visual cortex is the first way station in the brain, if you like, which is concerned with vision, in which visual goings-on, optical play, are manifested in the activity of the neurons. Now, there's something weird about the way this has been hooked up. If you look at that curious anatomical diagram in which we seem to have sliced a person's head through the middle, you'll see the eyeballs are at the front and the primary visual cortex is at the back. This is not a mistake. It is this way. And we'll look more closely at that plumbing in just a moment. But before we do so, let's have a look at the sensitivity of the retina. And we note that we are immersed in electromagnetic radiation at all times. The electromagnetic spectrum is vast and extends from very short wavelength to very large wavelength forms of electromagnetic activity. These include a lot of things that we often think of as separate or discrete. So modulation of the electromagnetic spectrum underlies radio transmission, television transmission, radar, X-rays, gamma rays, and the like. Only a tiny portion of the electromagnetic spectrum corresponds to what we call visible light, and this is in no way physically distinguished. Visible light is not something that pre-exists in the world before there are animals equipped with visual systems. Rather, visual light is just that narrow band of the electromagnetic spectrum to which our eyes have evolved to be sensitive. Now, if we look at the relative intensity of radiation coming from the sun, and we compare that with the sensitivity of the retina to electromagnetic radiation, we find that there's an almost perfect match. This is no accident. Our eyes evolved in a terrestrial environment. All eyes of all life forms that we know of evolved in a terrestrial environment in which light is present because we happen to be on a planet that orbits a star. That star has a particular composition. If the star had a different composition, we would have different eyes, clearly. Furthermore, the fact that there is light around and it bounces off things means that any biological innovation that can make use of that distribution of light for the services of the organism will be favored in the process of evolution. And so it is that our visual system is very finely tuned to the somewhat contingent nature of the radiation emitted by our sun. Now, this is the eye. You've probably learned a lot of this in secondary school. The eye is filled with vitreous humor. It's got the iris and the pupil on front, and the retina is at the back. I'm sure you know the basic anatomy of the eye. Sometimes the eye has been likened to a camera. We will, in a few slides, we will consider that analogy and whether it's appropriate. One thing to note here is that the retina is not a homogeneous surface. It's not just a blank sheet of paper onto which images are projected. It has a very specific structure. One part of it labeled there called the fovea represents the point at which the incoming light is most intensely focused, and that corresponds to the direction straight ahead of you at which you are looking in the world. So there's a geometric relationship between the fovea and the world. As we'll see, there are physiological distinctions around the fovea as well. Also, there's a nerve coming out. You've got to hook the eye up to something. The eye is not going to do any seeing on its own. So the optic nerve leaving from the back of the eye there introduces an irregularity into the surface of the retina, and there are no photoreceptive cells at that point. So even from here we can see that the retina is not a screen onto which images are projected. Now we find different kinds of receptors in the retina that are sensitive to different properties of transient light structure. We call them rods and cones. And shown here is the distribution of rods and cones in a cross-section across the retina. At 0 degrees here is the fovea. So this is a cross-section that happens to include the fovea and the blind spot. The blind spot, just to the right of center at about 15 degrees on the nasal side, has an absence of both rods and cones. Most of the retina has just rods. There's about 150 million rods in one retina. They are broadly distributed, and rods are sensitive to very different things than cones. Rods are sensitive to very low illumination levels. Rods do not make any color distinctions. I won't say that they see in black and white, because seeing is not something rods do. Seeing is something people do, and we're going to keep emphasizing that. But the rods have no sensitivity to what we consider color. The cones have a very narrow distribution, and they all are distributed, nearly all are distributed around the fovea. As I said, the fovea is geometrically the point that corresponds to where you're directly looking. That means this is where you're attending, and this is also where you see clearest and where things are most in focus. Cones contain photosensitive receptors which do differentiate among what we call colors, although the relationship between the receptors and the colors we perceive is extremely complex. But you can clearly see that the retina is anything but a blank sheet of paper. It is a part of an active organ with a very defined and very contingent structure. There's only about 7 million cones compared to 150 million rods, and we have about 1 million nerve fibers from the optic nerve heading towards the primary visual cortex. Now let's look at that weird plumbing again. We're looking down from above at a cross-section through the head. The eyes are at the front, and you can see the optic nerves leaving both eyes. And then we reach a point called the optic chiasma, in which some fibers cross to the other side and some don't. We'll see the consequences of that now in just a minute. There are then several way stations at which one nerve meets another in a synapse, sometimes splitting the message, if it's a message, sometimes splitting the pathway, and inducing some kind of transformation. So it takes about 3 or 4 synapses before we arrive at the bottom here at the primary visual cortex. Now, the traditional discourse around the senses would lead us to have that the eyes let in the light, that the ears let in the sound. That suggests that these are input systems. Now, it is possible to treat them as input systems, but that is no guarantee that that is going to be the most useful way to see them. There are paths from the eyes to the brain, but there are paths from the brain to the eyes, to the muscles around the eyes, and pathways that obviously orient the entire body, for as the body moves, so what you see moves. When we think of sensory modalities as input systems, we've made a mistake. The relationship of the body to the world is not unidirectional, and every time I move, so what I see changes. Heinz von Förster, who we met in a previous lecture, used to joke that we see with our legs. And he's absolutely right. We see with our legs just as much as we see with our eyes. As I walk around, so what I see changes. The eyes are not doing the seeing, and nor are the legs doing the seeing. The person is doing the seeing. Now, back to this strange anatomy, and let's start off just by noting the coarse structure of the outer surface of the brain, the neocortex. The brain is, of course, bilaterally symmetric. It has 2 halves. Each half can be distinct. We can make out 4 large areas on each half of the neocortex. The frontal lobe occupying basically the front half of the brain. The temporal lobe at the side, just behind the ears, although we'll find the plumbing in the ears is no more sensible than the plumbing in the eyes. The occipital lobe is out all the way at the very back, and that's where we're going to find the primary visual cortex. And between the occipital and the frontal lobe sits the parietal lobe here, somewhat at a remove from immediate sensory surfaces. Now, if you turn your attention to the figure in the top left, you're seeing a cross-section through a head looking at a spoon. The spoon is, as we're in the visual field. And there, the paths that cross and the paths that don't cross are shown. That is, from each eye, we see a red and a blue line going out. Those correspond to activity originating in the right side and left side of the retina of that eye. After the crossing over, half the fibers in each case crossing, we see a pure blue line and a pure red line. What this means is that the relationship of the right half of the visual field is affecting the transmission of optical information on the left side of the brain. Likewise, the left half of the visual field projects to the right half of the brain. It's not the case that the left half of each eyeball projects one way or another. The distinction that is captured in this crossover makes a rather more sense. What the distinction is that the right half of my visual field, when I look out, that right half of the world is processed initially on the left side of the brain and vice versa. So this is very, very strange plumbing indeed. It's complicated. The eyes are very close to the brain, but they don't contact the brain there. That's the way biological systems evolve. Now, what we're looking for, however, is the mapping. We want to understand the spatial mapping between the layout on the retina on the one hand and the layout on the primary visual cortex on the other. Now we'll simplify this. This is a schematic diagram of a monkey retina on top and brain at the bottom. And this brain, you're looking at the back half of the brain that has been opened up. Let me do this like this. Opened up so that the areas that you see on the right-hand side actually fold over to meet the areas on the left-hand side. You're looking, in other words, into the structure of the occipital lobe. Now on the retina, we've identified three broad areas just to be able to talk about its spatial layout. The fovea in the middle, which has areas 1, 2, 3, and 4. Then a perifoveal area around the fovea, 5, 6, 7, and 8. And then a peripheral area, 9, 10, 11, and 12. There's not a strict anatomical distinction between the perifoveal area and the peripheral area, but this will allow us better to understand what's going on when we look at the organization of the primary visual cortex. Moving down to the brain below now, we see the same organization. Those same areas are there. So the projections from area 3 in the retina go to area 3 in the brain. But notice the relative sizes. Those areas projecting from the fovea have lots of brain real estate given over to them. Those areas from the periphery have very little. The periphery, remember, is not involved in, has different sensitivities to the fovea. It's very sensitive to motion, to low light illumination, but not so great on details and color. So what's happened here is that we can see the differentiation of the functional importance of these different areas of the retina is elaborated in the brain. There are distortions that occur in projecting from the retina to the brain, but they are principal distortions. We don't get chops and cuts, rather we get a topological squeezing and stretching. The retina stretches out to occupy a larger area in the brain, while the periphery shrinks to occupy a smaller area, as if we were dealing with rubber sheet topology here. So having looked at that and considered what we've just said, there is some reason for pause for thought here. We said, and I'm sure you were taught in school, that the eye is like a camera, and there are certain reasons why that analogy might spring to mind. So the laws of projective geometry apply to eyes and to cameras alike, and they are constructed with the means to exploit optical geometry, and they both make use of a lens and an aperture. The camera, however, captures an image on film or on a charge coupled device, and you do with the image what you like. The eye does not capture an image. That may be a surprise to you. Vision has very little or nothing to do with images. Images belong in a different discussion. Eyes do not record images, and images are not sent to the brain. Cameras themselves, of course, don't see, nor do eyes, but eyes are integrated into a larger visual system, which is the entire body. As we said, we see with our legs. Seeing is done by the person as a whole, and seeing, as we'll find out, is purposive. Remember the box jellyfish larva, which had the simplest visual system that we could identify. It revealed its purpose very clearly. It was to steer the little animal towards the light where food is. Now I said images are not being sent to your brain, and we need to draw a line here. The study of images is a philosophically extremely rich topic. Images are instantiated in objects in the world. So you might come across a photograph, or indeed you're looking at a slide right now. But what you see is complicated. It's hugely influenced by your entire personal history, and longer, by your culture, by your education, by your experience of other images. One very good writer on the topic, Ernst Gombrich, pointed out that there is no such thing as the innocent eye. You don't see on the basis of some image which someone else will interpret. Rather, seeing is an interpretive act. It is a means of understanding. And here we see the point to sense making. So if you see with your full understanding, which itself has a really complex history and is culturally specific and has its own history, then it makes no sense to think of vision as simply the projection of images for analysis by someone else. We'll wrap up this video with just a painting of this notion of the innocent eye. So What are you looking at now? What are you seeing right now? Well, what you're seeing is a slide on your computer screen. On that, there is an image. It's an image, a photograph of a painting, another image. That painting itself is a painting of a bunch of men and another painting. So we've got images within images within images, and a cow. And what are these gentlemen doing? Well, on one reading, they are presenting the cow with an image of a cow in order to test the hypothesis of the innocent eye. That painted cow looks to us like a cow. The question is, will it so resemble a cow to a cow? But the naivety of the question, the idea that that might make any sense, is revealed when I ask you, what are you seeing? You're seeing your computer screen, on which is a photo, on which is a photo of a painting, and a bunch of other stuff. So as you can see, when we start to think about what an image is or where it is, things get very, very complicated, and we cannot separate the seeing of an image from the interpretive framework with we bring to bear on it.